Look at the reality of standard cancer care. Modern, highly targeted chemotherapies impose a staggering financial toxicity on patients, routinely exceeding $111,000 a year, while demanding a devastating physical toll from the body. Now look at generic, off-patent antiparasitic agents like Ivermectin and Mebendazole. These compounds cost pennies by comparison, and over the past decade, independent preclinical research has revealed they possess significant anti-cancer properties. They operate through a multi-targeted biological mechanism. Mebendazole physically breaks the microtubule scaffolding that cancer cells require to divide. Simultaneously, ivermectin disrupts multiple signaling pathways, starving the elusive, therapy-resistant cancer stem cells responsible for tumor recurrence. Yet these drugs remain trapped in what economists call the profit void. Because they are unpatentable, multinational pharmaceutical companies have zero financial incentive to fund the massive, large-scale clinical trials required to bring them into mainstream oncology. Patients navigate a medical ecosystem that willfully ignores biological efficacy if it lacks economic profitability. This structural vacuum leaves desperate people searching for alternatives, creating an environment ripe for exploitation. This chart displays a publicized 84.4% clinical benefit ratio for cancer patients on an off-label ivermectin protocol, but it hides a structural failure, excluding a 38.1% dropout rate. Erasing missing subjects engineers a massive positive responder bias. This workflow diagram reveals a fatal epistemological flaw. The outcomes were captured entirely through unverified, voluntary digital surveys. The researchers reviewed zero medical records and required zero objective radiographic scans to substantiate the claims of tumor eradication. The study also failed to control its variables. Nearly a third of the patients claiming a clinical benefit from these generic pills were simultaneously receiving highly potent, standard-of-care chemotherapy and radiation. It is scientifically impossible to isolate the drug's effect from the conventional treatments. The final collapse in credibility lies in the commercial disclosures. The primary authors are directly affiliated with the exact telemedicine company selling the medical consultations and dispensing the compounded prescriptions being evaluated. The result is disguised marketing, not empirical science. Desperate patients are manipulated by the very alternative providers claiming to rescue them from the medical establishment. Faced with alternative medicine grift, we typically look to mainstream regulatory bodies to provide an empirical safety haven. Yet independent genomic researchers analyzing commercial vials of synthetic mRNA vaccines discovered massive levels of residual plasmid DNA contamination, vastly exceeding established regulatory safety limits. These vials contain undisclosed SV40 enhancer sequences, which act as highly active nuclear transport signals. When encapsulated in lipid nanoparticles, these DNA fragments pose a theoretical risk of insertional mutagenesis, acting like corrupted foreign code actively transported into the human operating system, where it can inappropriately activate oncogenes or disrupt delicate tumor suppressors. Independent molecular analysts describe this dynamic through the hybrid harms hypothesis. Forced, prolonged production of synthetic spike proteins creates a persistent, highly inflammatory toxic milieu throughout the body. Over time, repeated antigen exposure drives a systemic immunological shift known as IgG4 class switching. The immune system's cytotoxic guard dogs are put to sleep, creating an environment of immune tolerance that allows latent, aggressive cancers to bypass normal immunosurveillance. Pathologists are also documenting systemic hematological anomalies, long, fibrous, amyloid-like blood clots forming in the vasculature. Unlike normal clots, these structures physically resist fibrinolysis, the body's natural enzymatic process for breaking down clots. Regulatory agencies have systematically dismissed these severe biological anomalies. The refusal to investigate these safety signals demonstrates that multinational financial capture has severely compromised objective global health oversight. We are caught in the retreat from reason. Patients are trapped in a deadly crossfire between the financial dogma of captured regulators and the unverified claims of alternative medicine profiteers. One faction manipulates digital survey data to sell off-label supplements, while the other suppresses genomic contamination realities to protect multi-billion-dollar biologic profits. Resolving this crisis requires the complete, legally enforced decoupling of the pharmaceutical industry from public health oversight bodies. We must demand a rigorous return to independent empirical testing. The exact same evidentiary standards must be applied to a $2 unpatentable generic compound as to a novel population-scale mRNA platform. The era of managerial dogma and science by press release must end. True scientific integrity relies exclusively on one thing: transparent, unmanipulated raw data.